Welcome back to my course Aspects of Biochemical Engineering, and we are last couple of lectures we tried to discuss some problems that are associated with this cell growth kinetics. we tried to solve several that numerical problems, and we discussed that initially we discussed the batch process, then how you can monitor the cell mass concentration and substrate concentration at different times. Then I try to discuss that the fed batch process, how you can find out the cell mass concentration, how you can increase the cell mass concentration in the fed batch reactor. And then in the last lecture I did some detailed analysis on the chemostat process, because chemostat appears to be the most advanced process as per biochemical industries are concerned. Now this lecture we want to, because last lecture if you look into that, mostly we gave stress on the substrate conversion with respect to cell mass formation, and maybe other than cell mass, that is the product formation. But here we also considered another very interesting term, what you call maintenance of the cells. That I told you that part equation that deals with the maintenance of the cells, that when cells we keep it in a particular culture, they move one place to others, they require some kind of energy, not only for cell when cells undergo some kind of rupture, for the repairing of the cells we require some kind of energy. So all this that how the maintenance energy is used. So these problems will deals with that part. Let us see how we can solve this problem by using all the three factors. Now we have come across three factors. One is your cell mass formation, your product formation, and the maintenance of the cell. So substrate is going for all the three purpose. Now the first problem, if you look into, this is the Zymomonas mobilis. I told you Zymomonas mobilis is kind of bacteria. Now mostly the alcohol ethanol fermentation take place by using yeast cells, the Saccharomyces cerevisiae. Now scientists they are working very hard how this Zymomonas mobilis can be used for ethanol fermentation, because doubling time of Zymomonas mobilis is much less as compared to the yeast cells. Now this Zymomonas mobilis is used to convert the glucose to ethanol in a batch fermenter under the anaerobic conditions. The yield of biomass from the substrate is 0.006 gram per liter. But here I want to just point out that if you look at the aerobic conditions, the yield of biomass is very high, maybe 0.3 to 0.5 grams per grams. But anaerobic condition, it is usually we have very less amount of cell mass formation. That is why this here it is 0.6 gram per gram. The product yield, the yp by x equal to 7.7 gram per gram. That means 7.7 gram of ethanol produced per gram of yeast cells, gram of bacterial cells. The maintenance coefficient is 2.2 gram per hour. That and specific rate of product formation for maintenance is 1.1 hour inverse, and the maximum specific growth rate of Zymomonas mobilis is 0.3 hour inverse, and 5 grams of bacteria is inoculated in 50 milliliter media containing glucose 12 gram per liter. Determine the batch culture time required to produce 15 grams biomass, achieve the 90% substrate conversion, and produce that 100 grams of ethanol. So that the problem has three different parts. One is you have to find out, to get the 15 grams per liter grams of biomass, how much time is required in a batch process. Then you have to find out that to achieve the 90% substrate conversion, how much is, that time is required. Then to produce 100 gram of ethanol that how much time is required. So this I consider this a very interesting problem. Let us see how we can solve this problem. Now, as I mentioned before, that whenever we want to solve any problem, it is better to write what is the given data is given. What are the given data? Y x by s, that is what we call cell mass yield is 0.06 gram per gram. Y p by x 0.77 gram per liter. Maintenance coefficient is given. Mu max is given. MP is the maintenance for the product formation. X0 value is given 0.5 gram per 50 milliliter and S0 is 12 gram. Now we know that in the batch culture this already we derived equal to ln x by x0 by mu max. Now x0 value we can easily calculate. The 5 gram was the initial cell mass concentration. Your culture volume is 50 milliliter. So your cell mass concentration is coming around 0.1 gram per liter, am I right? And final cell mass concentration is 50 gram, so divided by 50, that 0.3 gram per liter. So here in this problem we have x value, we have x0 value, we have mu max value. So if you put all this value, you can find out what is the time required for the batch process. Now if you look at the second part, that this is the first part is to what is the time required to get 50 grams of biomass. Second part is that what is the time required to get the 90% substrate conversion. Let us see how we can solve this. Now for doing so, first we shall have to do the substrate balance. Now what is the substrate balance? Rate of input equal to rate of generation plus rate of output, rate of consumption and accumulation. Now since this is a batch process, rate of input and rate of output, that should be equal to 0 and generation also equal to 0, am I right? So what we will be having, that we have rate of substrate growth and rate of product formation and maintenance into V. That as I pointed out before, we considered only the cell mass growth, am I right? Now we are considering 3 portion, that is the substrate is used for growth of the cells, one part is going for the product formation, here is the ethanol, another part is utilizing for the maintenance of the cells. So you have to consider all the 3 parts, and then this is the accumulation of the substrate. So this I can write like this, that what is the ds by dt for cell growth, how you can calculate. That is, I told you the ds by dt is equal to what? This is ds by dt. I can easily write that ds by dx into dx by dt. I showed you before also, just repeating. This is 1 x by s, and this dx by dt is mu into x. The x I have taken common. So mu into x divided by y x by s, that will give you the substrate required for the cell growth. Now similarly for the product, we can also substrate used for the product. I can write the similar way, the dx by dp into dp by dt. I can write this. So we that qp what is qp? Qp is the specific product formation rate. What is specific product formation rate? 1 by x dp by dt, am I right? dp by dt. Now dp by dt. Now divided by yp by s. Yp by s equal to dp by ds. The dp will cancel the ds by dt. And if you multiply it by x, then we will get the substrate required for the product formation. Similarly, we can find out the substrate required for the maintenance of the cell. If you multiply by x, you will get the substrate required for the, because we know that rate of substrate that is utilized for the maintenance of the cells is proportional to what? Cell mass concentration. So rate of substrate required for the maintenance will be equal to what? Equal to m into x. That is, the x is the kind of constant, what you call Michaelis maintenance coefficient. This is maintenance coefficient. So this equation, how it has come, and and we have already calculated what is the x value. How you can calculate x equal to x0 e to the power mu max into t. We have shown you before, I am not showing you again here. So, this is what we can put it here, and then this is equal to ds by dt. By rearranging the upper equation, we get then we'll get this equation, final equation, we will get this equation, rearranging all this equation. Then if we solve this equation, finally we will come in this equation e to the power mu max into T b equal to S0 minus S 1 by x by s qp y p by s mu max ms by mu max into x0 plus 1. This is what we can find finally, but you can I hope you can solve by yourself and find this equation. Now, then from that we can if you look at this equation, this you can now you divide by T b, you can easily calculate, you can T b equal to 1 by mu max, that ln this value. Then for 90% of substrate conversion, that what is the S value, that 10% will remain, that 1 gram per that if you see that what is the S value is given 12 gram per liter, am I right? 12 gram per liter. So, that so here we have done little mistake. This is no mistake, because this is actually that S0 is 12, am I right? And this is equal to 0.5. So, you multiply that 1.2, this is okay, this is not a mistake. And then we put the value, different values are given in this problem, and QP I can assume there to be constant, because now in this problem if we assume this this is equal to cell mass growth, and when there is a in this equation you have 3 part, am I right? When you have this is the cell mass growth, and another is the product formation and the maintenance. Since ethanol produced is coupled with the energy metabolism, then we considered QP equal to 0. And in that case, the Tp will be equal to 5.68 hours. Now, rate of product for change formation can be written as the dPV by dt equal to Qp into x into V. V is constant, that dP by dt, this we can write, this is x again we can write in this form, into V. We can Vb will cancel, am I right? And then we rearranging this equation, we will find this equation, and dP will be integrating from P0 to P, and this will be the final equation, and we will get this equation in this form. Then Tb equal to coming in this form, dB equal to this form, this is coming. Then now the specific rate of product formation can be this Kp equal to this form, then y p by x into mu max, because I can show you that dP, this is dP by dt, this specific rate of equal to, this is what y p by x. Y p by x means dP by dx, am I right? dP by dx, and this is mu equal to 1 by x dx by dt. So, this will cancel, so we can write specific rate of product formation, and this is the maintenance that is required during the this product formation. So, we can write this is the specific rate of product formation is like this, and then we have this equation, and putting these values we will get the QP equal to 3.41 hours. To achieve the 100 gram of this ethanol, the P equal to what? 100 divided by 50 milliliter is the culture, the concentration will be 2 gram per liter, P0 equal to 0. Then TB equal to, I can put this equation. In this equation, if you put, it is coming around 3.38 hours. So after 3.38 hours, you will get the 100 grams of ethanol. So all the three problems we can solve. Now in the next equation is little bit simpler. I hope you understand this. The steady state substrate and biomass concentration in a chemostat is given. S0 is this, and calculate the mu max and Ks and growth yield coefficient, and this is the true yield coefficient y dash x by s, and the maintenance coefficient. Now up till now we didn't discuss that how to determine the true growth yield. Now what is true growth yield? Up till now we come across the overall growth yield. Overall growth yield means gram of cell produced per gram of substrate consumed. Now when we say gram of cell produced, it may comprises of both the viable cells and the dead cell. But that when you do, or that, but that amount of cell mass that is produced, during that a part also go for the maintenance of the cells, and also a part goes for the product formation also. So, but when you say that we consider the true growth yield, then the product only goes for the cell mass formation, because this is not used for maintenance or product formation. That is called true growth yield. So question comes, how you can calculate the true growth yield and the maintenance coefficient of this we have not discussed yet. That let us see how we can solve this. And another very interesting thing is that up till now the cell growth kinetics we try to estimate by using the batch process. Batch process we find out that the first problem we solved that at different times we have cell mass concentration, we have substrate concentration, then we from that we find out the mu value, we find out the 1 by mu and 1 by s value, we plot the Lineweaver-Buck plot, and we determine that Ks and mu max value. Now same the question comes, is it possible to estimate the kinetic constant by using chemostat? The answer is yes. But now how it can be done? Because that Monod equation, we know mu equal to mu max is Ks plus S. Am I right? Now under steady state condition and sterile feed, mu equal to D. Now if mu equal to D, I can write in the Lineweaver-Buck plot, this is 1 by D equal to Ks by mu max 1 by S plus 1 by mu max. Am I right? So now you can easily plot 1 by, in place of 1 by mu, I can plot 1 by D versus 1 by S. So you can have a straight line. From the slope we can find out Ks by mu max, and for intercept we can 1 by mu max. So this we can easily find it out. Now this is exactly what is done here. This is the Monod equation that we have, and under sterile condition and steady state mu equal to D, then we can write in this form. Then we have 1 by S value and 1 by mu value. We can take in a graph paper, simple centimeter graph paper. We can plot this, and from the intercept we can find out the value of 1 by mu max, and from the slope we can find out the value of Ks by mu max, and then we can find out both the value of mu max and Ks we can calculate from this plot. Now question comes, how you can determine the maintenance coefficient, because that is important. So for maintenance coefficient, if you look at the part equation, what is the part equation we have seen? That is 1 by y x by s. This is overall. Am I right? This is equal to 1 by y x by s. It is true growth yield. True. true growth yield means I told you that when substrate is used for only for the cell mass formation, this is into m by mu. Am I right? Now, if you plot y x by s overall versus 1 by mu, then you will get a straight line. Slope will give you the value of m. Am I right? And this intercept will give you the value of y x by s true. True growth yield you can easily find. What you call, sometimes we consider as a y x by dash y x by s dash. This is true growth yield. So, we can find out these values. Now, this equation exactly we have written in this form. This is 1 by x, this is like this, and this mu we replace by d, and then we can have this table. We can write this table from this data that we have from the data. We can write this table. And then we can plot. Here we have the plot of 1 by y by x versus 1 by d. If we plot it, then we will get this intercept. Slope will give you the value of maintenance coefficient, and intercept will give you this. The maintenance coefficient will be 0.0019 hour inverse, and yield coefficient will be 0.5. This we can easily, and this yield coefficient, this is called growth yield. Growth yield is what we call true growth yield. When substrate is used only for cell mass growth, it is not used for neither maintenance nor the product formation, then we call it true growth yield, and this remains always constant. For a particular organism, this should remain constant. Okay. Now, next problem, I hope that this problem is the typical problem, that two chemostat system is used for producing the secondary metabolites. We know the secondary metabolites that usually most of the secondary metabolites form during the stationary phase. You know that, and the volume of the each reactor is 0.5 cubic meter. 0.5 cubic meter means how much? 0.5 cubic meter means how much? This is 1 cubic meter is 1000 liter. So 1.5 is equal to 500 liter. Am I right? So, the flow rate is 50 liter per hour. The mycelia growth occur in the first reactor, and second reactor is used for the product synthesis. The concentration of the substrate in the feed is 10 gram per liter. The kinetics and yield parameters of the organisms are y x by s, k s, mu max, q p, and y p by s is given here. Assume the product synthesis is negligible in the first reactor, growth is negligible in the second reactor. Determine the cell and substrate concentration entering in the second reactor. What is the overall substrate conversion? What is the final concentration of product? So what is the problem? Because let me explain the problem so that the problem is like this. We have two reactors. This is 1 and this is 2. This is like this. So, we have a starter here, and here we have a starter here. Am I right? Then this is liquid, and this is coming like this, and this is the starter, and here this volume is 500 liter, and this is also 500 liter. Both capacity is 500 liter. Now, here only the cell formation is there, and here only the product formation is there. That is the assumption we have. So, we can assume here cell formation. So, here this is x 0 and this is x. So, whatever x is there or x 1, and here also it is x 1, because we assume that no cell mass formation is there, and here also there is no here P0 here, this is also P0. So, here will be P1, only the product formation is there. So, this is the how this problem that we shall have to deal with. So, what are the parameters are given? What are the parameters are given? This volume is 0.5 cubic meter, flow rate is 50 liter per hour, S0 is 10 gram per liter. 10 gram per liter we can write 10 kg per cubic meter, and Yx by S is 0.5 kg per kg. Ks is 1 kg per cubic meter, mu max is 0.13, Qp is given, and this is given. Now, what is the dilution rate? I can write this is the flow rate 50 liter per hour and 500 milliliter. So, 0.1 is the dilution rate. From that we can easily find out the S1 value. S1 value is equal to Ks into D divided by mu max minus D. So, this is 5 kg per cubic meter. Similarly, we can find out the cell mass concentration if we assume x0 equal to 0, this equal to 0. If we assume, then we can find out x1 value. It is coming about 2.5 kg per cubic meter. Am I right? Now, so in this problem, the second reactor, it is assumed that no cell growth. I can write x1 equal to x2, that is 2.5 kg per cubic meter. The substrate balance in the second reactor, how we can write? The rate of substrate input F into S1, and rate of generation will be 0. Then rate of substrate output F into S0, rate of substrate consumption into volume. So, this we can write in this form, that the rate of product formation, and then we can write the S2 value. S2 value is coming around 3.3 gram kg per cubic meter. So, what will be the final overall conversion of the process? Initially, we have 10 gram per liter. Finally, it is coming 0.3. 10 minus 0.3 by 10, that is 97%. Now, the next problem is that what is the final product concentration? The product balance in the second reactor is F into P0, dP by dt into P into Fp. Am I right? Now, if it is like this, then the P0 we can assume to be 0, and then this equal to this, and these all values are given, flow rate and Qp, x2 and V value is given. So, we can easily find out final product concentration. It is not very difficult. So, in this particular lecture, what I try to analyze that in a particular biochemical process, that three things simultaneously take place. When substrate is consuming, that your cell mass formation is there, your product formation also there, and maintenance of the cells. Now, when all the three things are involved in this process, how you can take care to find out the different parameters in the system. So, in the previous lecture, we showed you how we can find out the kinetic constant like mu max and K's value. Now, in this particular lecture, I try to tell you not only K's and mu max value in the but also how you can calculate the value of true growth yield, also maintenance coefficient, and also you try to find out that how you can calculate the product for concentration in the particular reactor, final product concentration, how we can find out. Thank you very much.